The next thing that we want to study is the scientific method. The role of a scientist is to ask questions about the things that we observe, but the process of science is simply a method for gathering information and acquiring knowledge about the world around us. And there isn't necessarily a single formalized set of steps to follow for doing an experiment. However, all scientists generally follow what is known as the scientific method. It's just a way of learning about the world, about the natural world, by applying rules of logic to the way that information is gathered and the way that those conclusions are drawn. So the scientific method is a systematic, logical approach to gathering information and reaching conclusions. The first step in the scientific process, or the scientific method, starts with a simple observation about the world around us. However, for a scientist, it's not enough to just observe the world, but you also have to ask questions about the things we observe. So your observations should prompt a question. That question can then be formulated into a hypothesis. So that step is to develop a testable hypothesis or a possible explanation for the question that you've asked or the observation you've observed. We have to have a direction to travel in. The very best hypotheses are going to be clearly defined statements that we either want to confirm or deny. So think of it as a true or false statement. We don't want an open-ended question. It's kind of like a diagnostic test. When we're doing a blood panel, we aren't saying what is wrong with the individual. We break it into smaller chunks that we can either confirm or deny. Do they have this type of poisoning? Yes or no? Is there an elevated presence of white blood cells in the blood? Yes or no? Is there high alcohol content? Yes or no? We break it down into very easy to observe, quantifiable conclusions or observations that we can either confirm or deny our hypothesis. So once we have formulated our hypothesis, we then need to make a prediction. The prediction of the hypothesis helps us understand our hypothesis. It helps us know what we are looking for. Now, it shouldn't temper or flavor your results in any way. Your results should be objective and unbiased, but having a prediction should give you a general idea of the validity of your hypothesis or the validity of the test, your experiment that you are going to design. So for example, later this semester, we are going to be studying various food groups. We're going to take an item of food and we're going to break it down and test it for certain components. So for example, you may take peanut butter. Now, if we were going to study peanut butter and test it for the presence of simple sugars, we should be able to make our hypothesis. My hypothesis is that, yes, peanut butter contains simple sugars. That's a simple statement, and we're going to either confirm or deny that there is simple sugars in my peanut butter. Now, I'm going to take a step further and make a prediction. I think that peanut butter is probably going to test positive for simple sugars. Just because reading the back of the label says there's a lot of sugar in there. I know peanut butter is very sweet. I'm expecting there to be a lot of sugar in there. Then I'm going to run my experiment, and I'm going to see is there sugar present in peanut butter. And if I run my results and it says that yes, there is sugar in that peanut butter, I can say, okay, this experiment confirms the hypothesis. If, however, I run the experiment and I see that there is no sugar present in peanut butter, I go, that's interesting. I'm a little surprised by those results. I'm going to run this experiment again. And what we're going to find is that the best experimental data comes from collecting large amounts of data, not just running the same experiment over and over again to get verified results, but then also to run other experiments. It is possible that the sugar test I ran is not very sensitive, or it is possible there's something wrong with it. So there are other ways that I could look for sugar, or there are potentially other tests that I could run. And so all we can confirm, and if I run all the tests and I cannot find any trace of sugar in that peanut butter, then I can say, okay, by this experimentation, by this process, I will have to deny my hypothesis that there was sugar in this peanut butter. So when we are setting up this experiment, we're going to provide a prediction, and then we're going to experiment with two groups, usually a control group and an experimental group, to determine whether or not that hypothesis is supported. As I said, we're going to make a prediction. But another thing that we could do instead of just, or in addition to making a prediction, is to run that same sugar test on a batch of sugar water. Now, should sugar water test positive for sugar? Okay, I left a pause there so you could answer, but the answer is yes. Sugar water that has sucrose in it should test positive for sucrose, for simple sugars. So what I can do is run this experiment twice. Once I run it and I test my sugar water for sugar, and once I run it again and I test my peanut butter for sugar. If my peanut butter results come back that there's no sugar in the peanut butter, I can say, okay, it did not test positive for sugar. But if my sugar test comes back, my sugar water test comes back, and it says there's no sugar in that sugar water, it might give me some pause to think, maybe there is something wrong with the test itself. So this is what we call a control. It's a group that has run along the experiment along the same lines to help us determine the validity of that experiment or the validity of that test. So in this case, when we are running an experiment, both groups should be treated identically, except for one independent variable, that factor whose effect the experiment is designed to reveal. So in the case of sugar water, I should put, if I'm going to put 2 tablespoons of peanut butter into my experiment, I should put in the same amount of sugar. So I'll put in 2 tablespoons of sugar into a half a cup of water, stir it up to dissolve, and then run my test. If that's how I'm going to test my sugar, then that's how I need to test my peanut butter. I am going to put 2 tablespoons of peanut butter into a half a cup of water, stir until dissolved, and then run my test. If I'm going to heat up my sugar water to help it dissolve better, then I need to heat up my peanut butter water to help it dissolve better. You're going to treat both groups exactly the same. The only difference is going to be the independent variable. In this case, the independent variable is going to be whether we use sugar or peanut butter. Once I have completed my assignment or my experiment, I should be able to reach a conclusion based on the results of the experiment and review statistical significance. So it's not enough to just determine, okay, was that a positive or negative result, but is that a valid positive result? We are not going to spend a lot of time in this class reviewing statistical significance. We're actually not going to spend any time at all on mathematical statistics, but you may spend some time on this in some other classes. But once we have reached a conclusion, we're then going to develop new questions and additional experiments to help refine our conclusions. I could say, okay, yes, there is sugar in peanut butter, but now I want to know, where does that sugar come from? Is it added sugar? Is it sugar from the peanuts? Is it a plant sugar? Is it fructose? Is it sucrose? Is it glucose? Where is this sugar coming from? So I could develop some new questions and additional experiments that are going to help further refine those conclusions. Over time, after many experiments have been repeatedly confirmed related hypotheses, a theory can then be developed that's going to offer broad-ranging explanation for some aspect of the physical universe. So here is just what that scientific method is going to look like graphically. So here we start off with an observation, something we observe about the world around us. We then need to formulate a question. Use that question to help us develop a hypothesis. That hypothesis, we use a prediction based on that hypothesis to help us develop an experiment or undergo further observation. Once we finish our experiment, we're going to gather our results and draw our conclusion. If our conclusion does not support our hypothesis, we go back to the drawing board. We're going to revise our hypothesis, make new predictions, test them out with new experiments, come up with a conclusion. If those results do not support our hypothesis, we go back to the drawing board. And so this is what science is. It's going to be repeated, formulating a hypothesis, testing your hypothesis, concluding based on the results, and then reformulating your hypothesis, testing again, maybe coming up with a new experimental design. you're going to see that 90% of anything you do in the scientific world is going to end in failure. If you're going to be a scientist, especially if you're moving into research, you need to become very comfortable with failure, because this is how we progress. This is how we move forward, is by going through this cycle of developing a hypothesis, coming up with a prediction, formulating an experiment, collecting results, drawing our conclusions, and then going back to the drawing board. Even if those results do support your hypothesis, you can then use those results to make new predictions, come up with new theories, refine that data, refine your experiment so you can draw more solid conclusions. So we're going to talk a little bit just about experimental design. We've talked about the process of scientific method as a whole, but now we're going to focus on that experimental design. An experiment is a series of procedures that are designed to test an hypothesis. That's simple. It's going to utilize deductive reasoning to make a prediction or an expected outcome. And the manner in which a scientist conducts this experiment is what's called the experimental design. When you are setting up or designing an experiment, there are several key features that all good experiments have. Number one, they have a control group. The control group is going to go through all the steps and all the stages of the procedure, but it's going to lack the one factor being tested. So, like I said before, when we are running our food analysis tests, we are going to use water as our control. If we if we test water for the presence of sugar, should we detect any amount of simple sugars in our distilled water? The answer is no. If we run our experiment and we come up with a positive result for sugar in our water, we'll know that there is something wrong with the experiment. And so just because if our water says that there is test positive for sugar, we cannot then draw any accurate conclusions when our peanut butter tests positive for sugar. If peanut butter tests positive for sugar, it means nothing if water is also testing positive for sugar. If everything is testing positive for sugar, then the results mean nothing. We're also going to use water as a control when we are testing for proteins. Would you expect that peanut butter is going to test positive for proteins? Yeah, we should theoretically see a positive presence of proteins in peanut butter. If, however, we also get a positive result for proteins in our water, then we can no longer trust the conclusions drawn from this experiment because there's something wrong. So that's going to be our control. It's going to go through all the steps of the experiment, but it's going to lack that one factor being tested. The independent variable is also known as the experimental variable. This is the variable that we are testing for. So in this example, it would be, this is going to be our peanut butter, our independent variable. So our control would be our water, our independent variable will be peanut butter, and our dependent variable, also known as the responding variable, this is going to be the change or the effect that occurs because of the experimental variable. In other words, in our independent variable, this is going to be peanut butter. Our dependent variable will be a positive or negative result for sugar. So our control is going to run through experiment, our peanut butter is going to run through the experiment, and what we are looking for is this dependent variable, that presence of sugar.